And I'll be putting this up online so that those of our friends that are not here today or yesterday, or if you guys want to review it again and see it, you're welcome to do that. So the first thing, let's kind of define where we're at with metabolism. It's all the chemical reactions that are occurring within an organism versus a metabolic pathway, which is going to be for a specific series of activities. So metabolism describes your cycles of wakefulness and sleepiness. It describes your ability to digest food and generate energy. It describes the breakdown of material so that you are able to produce proteins. It describes proteins that you are or are not able to produce. Everything that's happening inside of you chemically to make you function. But there are specific metabolic pathways that get to insulin, or that get to clotting, or that get to digestion of a specific food type. And we're going to start today by talking about the metabolic pathways. So to demonstrate this idea of small pathways, we're going to do a little word tricky exercise. We're going to start out with the word tread, and we're going to change one step at a time, one letter at a time, until we get to the word blink. I'm going to give you guys time to do this on your own, but keep in mind that you can only change one letter per step. And each new word, each new step, actually needs to be a word. It can't just be randomly changing, oh, I'll change the T to a V in this one, and then I'll change the R to an L, because that would make, read, bread. Oh, actually, that's a word. Huh? That would work. But you need to make sure that as you're changing it, the order in which you're changing it is going to get you to the next word and the next word. So don't just go randomly from the beginning of the alphabet, or the beginning of the word.
That's going to take a lot of time.
We'll give it a few minutes. Steps, changing just one letter at a time. We start out with tread. We go to bread, then breathe, then bleed, then blend, then blind.
Oh, I didn't do blind.
And then blend. So you were very, very close. Good. So the idea here is we have to have the same combination of steps to get there with those rules. Now, in some metabolic pathways, and you guys have seen this when we look at aerobic and anaerobic respiration, you can get part way and it could go this way or it could go this way. In this case, hi. Thank you. All right, so in this case, we have one pathway that goes through. If we were writing a story and we ended up with blink, that was the word we wanted to get to. But let's say halfway through the story, we stopped at bleed, we'd have a very different story. Here is a surprising story about a birthday party. Here is a horror story, hopefully not about a birthday party. Right? Or if we went to bread, we've got a cooking show or a cookbook instead. So depending upon, or even blend, we might have a cook show. But we're not making bread this time. Now we're making pancakes or we're making sauces. I don't know. So in every metabolic pathway, there is a series of steps, and you have to get to breathe to get to bleed. So each of the intermediate steps, you have to get there. So metabolic pathways can be disrupted. If we take out the ability to add another an L in here, if I say you have to get from tread to blink, but I'm not going to give you any L's, metabolic pathway over. You can get partway. You can get some intermediates. And if this could go in a different direction to a different metabolic pathway, that will still go. But you're not going to get any further on this particular pathway. So these metabolic pathways are super, specific. Now, today I'm not asking you to get specific. I'm going to give you this generic format. Generic because we're just calling it substrate 1 and enzyme 1. So think of substrates as the things that are going to react, and the enzyme is a catalyst that makes that reaction happen. We're going to talk in a minute about why it makes that reaction happen. So we start out with the stuff, and that stuff interacts with enzymes, and that enzyme produces some different stuff. And this different stuff, the product here, is now the reactant for the next reaction. And it triggers the release of a different enzyme. Not the same enzyme, which then produces other stuff, which becomes the reactant for the next reaction, which triggers another enzyme, which results in the actual thing that you were trying to get to. Now this is a nice, simple, straightforward one. I had somebody in my last class that went online and looked up just under images, metabolic pathways, and they found this thing that was just crazy. Yeah, you're looking right now and you're like, oh, you don't have to memorize those, it's okay. If you were to become a physiologist or an experimenter later in life and you were working with a metabolic pathway, you're going to know it. But memorizing pathways randomly is not really beneficial to you. So I give you this very simple straight line one. What are some examples of metabolic pathways that you can think of that we've already studied this year? Photosynthesis, which has some chain-like structures, but also has some cyclic structures. This is straight up from photosynthesis, right? Which cycle is this? Growth cycle. Excellent. So we've got our 6 carbon. I know you guys can't see the specifics from back there. You've got your 6 carbon, you've got your 5 carbon, you've got your 4 carbon. We've got decarboxylation happening here. Some of these materials will leave the cycle and new ones will need to come in, but some of these will just continue to circle around and around and around, accepting electrons, giving up electrons. So depending upon the cycle, it can be, depending upon the pathway, it can continue to cycle, or it can be a stopping and starting one. And this starting one over here, something has to show up inside the organism to trigger the beginning of that enzyme, or there has to be a demand for the end product, depending upon the cycle. And there's lots of variety, and they get really messy. So let's look at what's actually happening when we have this substrate 1 and enzyme 1 interacting. We have our enzyme right here, which has an active site, and has an allosteric site. Allosteric site means it's on the same molecule, but it's not at the same site as the active site, and it can affect the active site. We have our substrate here, and look at how nice and tidy it fits inside this active site, so that when they fit together, that will trigger the reaction to happen, or it will speed up the rate of the reaction, depending upon what kind of enzyme and substrate we're looking at here. We're going to look at in just a minute why that enzyme would affect the rate of the reaction or make that reaction happen. We're also going to talk about 2 different ways this reaction gets blocked, called inhibitors. Does anybody else need to sketch that, or can I move on? Okay. So, inhibitors, yes. So we have 2 kinds of inhibitors we can talk about, competitive and non-competitive. Inhibitors get themselves onto that enzyme and stop that enzyme from reacting with the substrates. That's the job of the inhibitors. We've talked about them with regards to a couple of different reactions. We have stimulants, things that will make the reaction happen, inhibitors that will slow that down. We're only talking today about these inhibitors. Now, the ways in which they can change it, we have competitive and non-competitive. Competitive means the inhibitor has a shape that will allow it to get inside this active site and sit there. It may not be a perfect match, so it's not going to trigger a reaction, but it fills that spot so that the substrate can't get in there, so the reaction can't happen. That would be a competitive inhibitor. Think about something like if I put a cupcake on the table and had people stand 3 steps back, 2 people stand 3 steps back, and then say go, and they race for the cupcake, one of them can get that cupcake, and then that cupcake doesn't exist anymore. So once this active site is filled, no substrate can get in there. It's either you get it or you get it. As opposed to a non-competitive inhibitor, let's use that same cupcake example. This allosteric site that the non-competitive inhibitor can sit into changes the quaternary structure of the enzyme. Because the enzyme is a protein, right? Remember when I made you do all that protein folding online, and you shift it just a little bit, and all of a sudden all these side chains clash, and it can change the shape of the active site, so that this little slightly folded differently, active site no longer accepts this wider, well, this hasn't changed, the substrate shape hasn't changed, but the active site got narrower, and so it won't fit in anymore, just because of this non-competitive. So if we think of that cupcake that these competitive people were racing for a second ago, in this case, the non-competitive inhibitor is not going to take the cupcake, it's just going to smash it. Bam! Hi! You didn't get it, and I didn't get it. Nobody's sitting in that active site, but the active site doesn't really exist in the same form anymore. I know, it's just mean to do to a cupcake. So it doesn't have to erase it, it just has to fit into this spot in the back. And this is a reversible thing. This reaction, if this non-competitive inhibitor goes away, the active site, if it hasn't changed too much, can change back. Same thing with the competitive inhibitor. If you have an inhibitor in here and it goes out again, now the space is available. So it's a way for the body to control, to release inhibitors, to control whether or not that reaction gets started or moves forward. And in some cases, it's the way for other chemicals that can get into your body to start or stop reactions that your body is not necessarily as much in control of. So, for instance, appetite suppressants, they'll go in there and they'll change the active site so you don't have that feeling of hunger. Or headache medicine can go in there and compete with, inhibit the pain receptors so that they don't convey that pain feeling. Or have you guys ever seen on TV commercials that'll talk about medications you can't take this with?
Yeah.
And it's because they're going to counteract each other. They're either going to be competitive or non-competitive inhibitors on each other. So if you take them both, you don't get anything. All right. So in words now, whoops, that didn't go away like I wanted it to. Just a second. Okay, now we got this. In word style then. So a competitive one, if you were describing it, it's going to get into that active site so that the substrate can't get in there. And non-competitive is going to sit in the allosteric site, which is on a different part of the enzyme. Could be on the back or the side. And you can have multiple active sites on proteins that different enzymes could react with. So your body could be efficient and produce an enzyme that has 3 active sites. And depending upon what substrate gets in there, it will drive different reactions. Get very complicated very quickly. And there's a different kind of inhibitor that we're going to talk about, which is an end product inhibitor. Oh, okay. You got it. So an end product inhibitor means that whatever the metabolic pathway's final product is, can actually act as an inhibitor to stop that process restarting. So if, for instance, going back to our word example, the word blink, if it could stop the changing of the words starting from tread, it would like sit over the top of tread and block the ability of that word to be changed at all. As a visual for this, because I think it's helpful to see it, we've got a metabolic pathway here with our cute little green substrate and our enzyme. The reaction of the substrate and enzyme 1 produces our second substrate, which is actually going to interact with a different enzyme. This product becomes a reactant to produce another substrate, which now becomes a reactant to produce this end product. And this is what the metabolic pathway is set up for. But guess what? That metabolic end product can go back into this original enzyme. There's an allosteric site that makes it change the shape of the active site. So now, this thing is saying, hey, we've made enough of me, stop the process. So this would be useful for something that's a short-term metabolic pathway. I need to process the milk that I just drank. Boom. As soon as I've made enough of that stuff, it goes back into the enzyme and stops the production of more enzymes. Why is this beneficial to the body to have the process stop more process? Yeah.
So you don't just like overflow the amount of like.
We've enough.
Yeah.
This takes energy. This takes raw materials to make. So you don't want to just keep cranking it out, right? If you were wrapping 200 gifts for somebody and you only have 150 friends, you don't want to keep wrapping gifts randomly. I don't know about you guys. I don't enjoy it enough to just sit there and wrap 50 presents that aren't going anywhere. I want to know that I'm done. I have enough. So the same thing can happen for short-term metabolic processes. There are some metabolic processes that are ongoing that are not going to have inhibitors on a good day. Processing oxygen, digestion, movement of nerves, and things like that. But when things are going well, they don't have this inhibition going on. But maybe some of you guys can think of either medications or drugs that people would take that would switch on or switch off processes so that they are no longer functioning the way they should. So this inhibition can work for you and against you. All right, so how is it that enzymes are able to catalyze these reactions? What is it they do? I think we've been through this before, but just to review and bring it into the concept of metabolic processes. We start out with this big, nice gray enzyme here, and there's some kind of a substrate that's made of multiple components. We don't have to name the components right now. But by getting into the substrate, by getting into the active site, this enzyme can end up straining the bonds that brings the two components together. Just by sitting in that active site, it can strain the bonds so that they break more easily, so that we end up with the components of that product, or of that reactant. What kind of a reaction would we describe this to be? You describe it as an A or a C. Catabolic, we're breaking it up. So enzymes can make a catabolic reaction happen at all, or happen more quickly, depending upon the pathway that we're on. So this reaction might happen, this breaking of the substrate might happen anyway, but it will happen faster with an enzyme aiding the reaction. The opposite looks just in the opposite direction. Let me show you that. If we have an anabolic process that is enzyme assisted, we have two separate substrates that are attracted to the active site, which brings them in close physical proximity, which allows bonds to form. If they stayed this far apart, bonds aren't going to form. So by physically bringing them together and they're attracted to the active site, now we end up with a product. So this is our anabolic reaction that is making stuff instead of breaking stuff. Questions? All right. So now we're going to figure out, why does this lower the activation energy, or what is the activation energy? Do you have any chemistry students out there that can answer that? What is activation energy? Let's get on that, shall we?
Like the ignition temperature for fires?
Yeah. It's that minimum amount of energy needed to make a reaction run. And in some cases, it might be thermal energy. It might be kinetic energy because you swirl the reaction. Or it might be enzyme assisted. So let me interpret this graph for you and then give you a second to copy whatever you decide you need to copy down. So we have this darker purple area up here that this shows without the aid of enzymes, this is how much energy would be required to make that reaction run. When the enzyme is present and it makes that reaction more likely to happen, this light purple shaded area shows us the area that the amount of energy that is required to make it run when there's an enzyme present. Because either we're having the substrate sit in those active sites and the bonds are more easily broken, or the active sites are attracting separate substrates and making that product more easily just by physical proximity. Why does that work? What normally does it take for two things to come together? It might take chemical attraction, right, and then electrostatic attraction. It might also, because molecules are constant and move, it might just be, hey, we're close enough. H2O, it wants to bond. As soon as those molecules get close, it's going to make stuff. It's going to make H2O. When we increase the temperature, what is it we're really doing to the molecules?
We're giving them more energy.
And that makes them do what?
They're more excited, they move faster.
Yeah, it just makes them move more. So that's why increasing the temperature might make a reaction move that isn't moving at room temperature, because all of a sudden those molecules, they're bouncing around and they're bumping into each other. And when they bump into each other, that chemical attraction then can take place. So you can see how putting them in enzymes and bringing them closer by using an enzyme can do the same thing. Just puts them in close physical proximity. Increases the odds that they're going to find each other and bond. And therefore it requires less temperature. Cool, yeah? Yeah.
It's like the whole activation energy, like when
take different amounts of activation energy. There are some reactions that will happen at room temperature. There are some reactions that will happen at colder room temperature. And there are some reactions that require extreme heat before that reaction is going to go. Or a lot of you can use electricity to stimulate those reactions instead of heat. Or you can use kinetic energy, depending upon the reaction. By swirling them up, you're now moving them physically around, so they have a better chance of bumping into each other. Any questions on this here diagram right here? All right, so we're going to summarize that with a few words there at the end. If you want to use those words, you can. It just talks about how the enzymes are changing that rate of reaction so that we don't need as much energy to go into it.